Today's lecture will be on the peripheral nervous system glial cells, and the first structure that we'll be talking about are going to be the satellite cells. These satellite cells, as pictured in this illustration, each one of these cells, they are going to encase or wrap around the periphery of each individual neuron cell body, or when you put a group of neuron cell bodies together, they form a ganglion. Their job is important because they are going to regulate the fluid composition or the environment around each one of these neuron cell bodies, including the exchange of nutrients and waste products associated with the neuron cell bodies. So in this illustration, we're looking at the posterior root ganglion, which is going to be important because it's associated with the sensory. So all sensory information will be traveling along these axons. They will be processed by the neuron cell bodies, and these satellite cells are going to be important in regulating the nutrients and waste products for the cell body operation. And then the sensory information will continue on into the central nervous system of the spinal cord. The next structure that we'll look at with peripheral nervous system glial cells are a structure called the neurolemmacytes. So the neurolemmacytes, also called Schwann cells, are associated with the peripheral nervous system axon. Their job is to, at the beginning, is to form a myelin sheath, which basically a myelin sheath is concentric rings of myelin that the neurolemmacyte will eventually put down to form this neurolemmacyte or area of myelination along the axon. In between each one of these neurolemmacytes or areas of myelin, you're going to have the neurofibril nodes. This is going to be important to allow for propagation of the nerve impulse or action potential along the axon. The importance of neurolemmacytes laying down myelin sheaths is to allow for faster propagation of a nerve impulse. When we're looking at myelinations of axons, it's important to understand what a nerve impulse is. So a nerve impulse is going to travel along the axon. It's a rapid movement of electrical charges along a neuron's plasma membrane. The speed and the impulse travels along the axon is affected by the process of myelination. This allows for faster propagation or traveling of the nerve impulse of the action potential along the length of the axon. So the first thing that we'll see is this neurolemmacyte, which has a centrally located nucleus at the beginning, is going to begin to wrap around or form in the second illustration these layers of concentric rings laying down myelination. So as this neurolemmacyte continues to wrap around the axon, you can see that the nucleus is starting to get pushed out towards the peripheral edges. Eventually, we're going to have consecutive layers of this neurolemmacyte that is going to form the myelin sheath. In step number 3, we begin to see, as this process continues, the neurolemmacyte continues to wrap around, and it is going to eventually, we're going to see the cytoplasm of the neurolemmacyte, and it's going to start to form this outer ring of myelin, which is the outer edge, which is a very delicate, thin outer membrane of the neurolemmacyte, which is called a neurolemma. So as the process continues, eventually what we're going to see is the wrapping is going to squeeze out the cytoplasm, and then eventually the neurolemmacyte and the cytoplasm of the nucleus are all pushed to the outside or the peripheral edge. And this is going to form this outer layer or the most peripheral layer of myelin called the neurolemma. With myelin sheathing, we see this both in the central nervous system as associated with the structure called the oligodendrocytes, and then we see the neurolemmacytes that are forming the myelin sheath in the peripheral nervous system. In the peripheral nervous system, these nodules or neurolemmacytes, they're called nodes of Ranvier, and these nodes of Ranvier or neurolemmacytes change in voltage occurs across this plasma membrane, so during the process of a nerve impulse traveling. So we can have, whenever we have myelination that occurs in the central nervous system of the oligodendrocytes, which are producing the myelination along the axons of the central nervous system, or the neurolemmacytes, we call this a saltatory conduction. And this specifically is associated with myelinated axons. And this is simply allowing for this nerve impulse to jump from one neurofibril node to another neurofibril node. Again, this is important. This node-to-node propagation allows for faster traveling of the nerve impulse or action potential. So axon regeneration, it also goes by another term called Wallerian degeneration or regeneration. And this is associated with the neurolemmacytes. So this illustration will specifically be talking about the neurolemmacytes, or otherwise known as the Schwann cell. So the first thing that we see is the effector here, the muscle tissue, and we see that there has been trauma that's occurred at the end of the axon. And so the first step in step one, the axon distally or further away from the injury site, so this location here, the first thing that we're going to see is the degeneration. So in step number 2, we can see that the axon itself is starting to break up, so we see that degeneration. And in step number 2, what we also see is the macrophages are going to come in and they're going to engulf or through phagocytosis, they're going to destroy the remaining portions of the axon. What survives is going to be the neurolemma. So again, the neurolemma is the outer sheath of the myelin, and this neurolemma is going to be important because it is going to be part of the process of forming a tubule. The other part that's going to survive the injury is going to be the endoneurium, and this area is important because this is where we see the individual axon is going to make its way through the injurious site and eventually attach to the effector. So in step number 3, as mentioned earlier, the only thing that survives is going to be distally to the injury is going to be the neurolemma and the endoneurium. And this is going to form this regenerating tubule. And what we're going to see in step number 4 is once we have the neurolemma and the endoneurium forming this tubule, you're going to see the axon begin to grow towards the effector site. So at this point during recovery, we still have not made contact to the effector, but we see that as this axon is going to grow, then we start to see the neurolemma site and the endoneurium start to enclose the axon, and that enclosure is going to allow for the beginning of this saltatory propagation or the action potential to jump from nodule to nodule through to eventually hit the effector or the terminal end site of whatever it may be innervating. So at this point we have function of the effector.